Oh, hey, what's up, everybody? Welcome to today's lecture. I'm going to do a couple of lectures right now that are about the rate of evolution. And one of them is about how fast evolution can be, and one of them is about how slow evolution can be. So this first one is about how rapid evolution can be. And to tell you just how rapid evolution can sometimes be, I'm going to tell you a story. And the story that I'm going to tell you is the story about the time that a tanager became a vampire. This is a bird from South America called the dull-colored grassquit. And this bird has a pretty broad distribution, mostly in western South America. It looks pretty unremarkable, just a kind of normal, boring, brownish bird that you might see out in your yard if you lived in Colombia. But this grassquit is really special because it belongs to a group of birds. Within that monophyletic clade is one of the most remarkable radiations in all of ornithology. The radiation that I'm talking about is a group of birds called the Galapagos finches. In this diagram, you can see 4 different Galapagos finches, and one of the things that you'll notice if you look at these finches is that they are incredibly different from one another in the shape of their beaks. Compare the bird in the top left, which is Geospiza magnorostris, to the bird in the bottom right, which is Camarhynchus parvulus, and you'll see that their beaks differ dramatically in length and width. And it turns out that the beaks of these Galapagos finches are adapted to their various diets. One of the most interesting things about the finches in the Galapagos is that even though these birds are really divergent from one another, they form a relatively young monophyletic clade. Look at this phylogenetic tree that was published in 2002 from Burns et al. This is a phylogenetic tree that was constructed from mitochondrial DNA sequences from the cytochrome B gene. You can see the branches of the tree there, and on the branch, each branch has a number. That number represents the support for that branch being correct in the analysis. And so, for example, if a branch has 100 on it, we can conclude that the branch has really good support in the data. Now, if we look at this tree a little bit, we can see our grassquit up here, obscura, and then look at this clade right here. Here's 100% Bayesian support value. That's really good support. All of these species in this part of the tree right here are Darwin's finches. And you can see that they form a monophyletic clade within this part of the tanager tree. What that means is that an ancestor of the Galapagos finches colonized the islands and then radiated into all of these really different species. In the Burns et al. Paper, they went on to measure the beaks of birds, both among Darwin's finches and their relatives and other genera of birds. And you can see those plots here. So each square is a plot for a different group of birds, with Darwin's finches and their relatives in the upper left, and then the other boxes are other genera as labeled. On the x-axis, we have beak length. So along here, we have short to long beaks, and then beak depth. So that's narrow to fat beaks on the y-axis. And here are the Darwin's finches in squares and their relatives in pluses, and you can look at all the other groups of birds here. The percentage in each box is the average percent mitochondrial DNA difference among species in that group. And you see first that Darwin's finches aren't really that different from one another in terms of their mitochondrial DNA. That's because this is a young radiation. But despite the youngness of this radiation, they span an incredibly wide range of beak shapes, from really short and narrow beaks to really long and wide ones. And in fact, the amount of this beak space that's occupied by Darwin's finches and their relatives is much greater than any of the other groups. Why might that be? Well, one of the reasons is that Darwin's finches, that's the genus Geospiza, and their relatives evolved much more rapidly in terms of their beaks than other tanagers. And why might that be? Well, if we imagine back to the origin of this group, the islands were colonized by an ancestral seed-eating ground finch. And the Galapagos Islands are volcanic, so they rose out of the ocean and then were colonized by birds and other animals, mainly from mainland South America. So compared to where those birds were coming from, the Galapagos Islands are really species poor. They didn't have a lot of competition. There were plants on the islands producing seeds and lots of open niches for these birds to evolve into. And you can see, looking across the Galapagos finches, that you have a wide variety of beak shapes, specialized towards eating insects, seeds, even buds and fruits, and parts of cacti, the cactus finch. And so from one ancestral seed-eating ground finch, you get this incredible, spectacular radiation of lots of different finches with lots of different beaks in a relatively short amount of time. The Galapagos finches were always known as a rapid radiation because scientists had observed this pattern, and even Darwin used Galapagos finches as part of his inspiration for the theory of evolution. But we didn't really know exactly how fast evolution could happen in the Galapagos finches until these scientists did their work. So this is Peter and Rosemary Grant in this picture. They are a couple who has been doing scientific research in the Galapagos for the last 40 years. And the Grants' work has really changed the way that we think about the pace of evolution. Let me show you how. Much of the Grants' foundational work was on this species, the large ground finch. You can see it's one of the species with a really particularly thick beak. And what the Grants did is they just went out every year. So these plots go from 1970 to the year 2000, so 30 years of data, and measure various aspects of body size, beak shape, and beak size from one year to the next in the Galapagos. So here you have two species, Geospiza fortis and Geospiza scandens. Body size on top, beak size in the middle, beak shape at the bottom. The y-axis is time, and the x-axis represents an index of that shape. What you see when you look at these plots is really an amazing amount of change. And follow-up studies that the Grants and their collaborators have done since this study have shown that a large portion of this change actually has a genetic basis. So it's not just phenotypic plasticity changing with the environment. It's actually genetic change. Furthermore, the Grants can show that these changes are related to changes in the environment, especially in terms of what seeds are available. So when there's lots of large, hard-to-crack seeds around, for example, there's selection for robust beaks that are really blunt, and vice versa. And by zooming in on some of these changes from one year to the next, the Grants were able to estimate rates of evolution that were much faster than people would have expected. So, for example, that little change right there that happened in the mid-1970s in body size of Geospiza fortis. So it's this rapid evolution that can help us understand how we go from one ancestral seed-eating ground finch, to this large radiation of Galapagos finches in a relatively short amount of time. We also have learned since the Grant studies about the genetic basis of these traits. So here's one example of a study by Lamicini et al. In 2015 showing an association between one genotype and an ALX1 gene and a score of beak shape, basically how pointed the beak is. So here birds with a PP genotype have a much more pointy beak than birds with a BB genotype. This series of studies shows us that evolution can be rapid. It shows that the mechanism of that evolution can be changes in the environment, causing differences in selection on body morphology, and we can even get down to the specific genes that are being selected for or against during those different times of environmental change. And altogether, these studies really give us a good handle on this process that allowed us to go from one seed-eating ancestral finch to the variety of finches that we find in Galapagos today. My favorite Galapagos finch, though, is this vampire finch. This is Geospiza difficilis. This finch, one of the things that it can do is land on animal hosts like this Nazca booby and gouge into their skin to obtain blood. And one of the things that this vampire finch gets from the blood is water. Water is really a limiting resource in some of these Galapagos islands. And so this finch has become a vampire in order to obtain the water that it needs. And it's just another good example of the wide variety of forms that you can get from one ancestor when you have evolution acting by natural selection. Galapagos finches are a great example of a phenomenon that we call in macroevolution an adaptive radiation. And adaptive radiation is defined as the evolution of ecological diversity within a rapidly multiplying lineage. Rapidly multiplying means really rapidly speciating here. So finches form new species rapidly, and those finches adapt to different parts of the environment, different niches. Another really good example of adaptive radiation is the anoles, and you can learn more about the anoles in 3 episodes of The Lizard's Tale, which I'll post for you on BBLearn. So the Galapagos finches are, I think, one of my favorite examples showing that evolution can be incredibly rapid. Next time we'll learn that evolution can also be incredibly slow.